Dear students, welcome to all of you. Today we are here to discuss structure of carpel and female gametophyte. First, the structure of carpel. The carpel is the gross morphological part of the flower concerned with female sporogenesis and gametogenesis and is the homologue of the stamen. The carpel is a leaf-like organ composed of the ovary, style and stigma. Carpels are the megasporophylls which make megaspores and their products, that is female gametophytes. A carpel resembles an involutely folded leaf lamina with its margins fused. The number of carpels is usually fixed for a plant species and it is described as monocarpellary, that is single carpel, bicarpellary, two carpels, tricarpellary, 3 carpels, tetracarpellary, 4 carpels, pentacarpellary, 5 carpels to multicarpellary, usually having more than 5 carpels. When the number of carpels in a gynoecium are 2 or more, they may be free or fused. When the carpels are fused with each other, the condition is described as syncarpous, for example crucifery, and when free, they are described as apocarpous, for example ranunculaceae. The syncarpy may involve fusion of only ovaries, for example dianthus, or ovaries and styles, for example hibiscus, or entire carpels, for example brassica and papaver. At the tip of the carpel is a sticky stigma that receives pollen. The stigma may be bifid, for example in compositae, dome-shaped in brassica, funnel-shaped in crocus, striate in papaver, or plumose in case of gramineae. A style leads to the ovary at the base of the carpel, which contains one or more ovules in the intra-ovarian chamber or locule. The ovule develops into a seed on fertilization. Depending upon the number of locules, the ovary is termed unilocular, 1 locule, bilocular, 2 locules, trilocular, 3 locules, tetralocular, 4 locules, pentalocular, 5 locules, or multilocular, more than 5 locules. In some primitive plants, for example Drimys, Digenaria, there is no distinction of ovary, style and stigma. The pollen is received on a longitudinal stigmatic crust extending from apex to the base. The whorls of floral organs, that is sepals, petals, stamens and carpels, are attached to a part of the stem called the receptacle. A single carpel or a group of fused carpels is sometimes called a pistil. The megasporangium consists of nucellus covered by integuments. The integumented megasporangium is called as ovule. The angiospermic ovule consists of a central body, the nucellus, and a basal stalk by which it is attached to the placenta, the funicle. The nucellus is protected and enclosed by 1 or 2 sheaths, the integuments. The opening in the integumentary sheath where tip of the nucellus is exposed is the micropyle. The basal part of the ovule where nucellus, integuments and funicle merge is called claza. In the nucellus is present female gametophyte, also called embryo sac. Now the structure of female gametophyte. The female gametophyte, also called embryo sac, is mostly a 7-celled structure. It has a well-defined boundary and is surrounded by a thin cellulosic wall. There is a large central cell with 2 polar nuclei which fuse to form the secondary nucleus. The micropylar end of the central cell is occupied by the egg apparatus comprising an egg cell and 2 synergids. The callosal end of the central cell is occupied by 3 antipodal cells. The cells of the egg apparatus and the antipodal cells are haploid, whereas the central cell is binucleate or diploid. The mature female gametophyte is a specialized unit with a variety of physiological and biochemical interactions between its constituent cells. All the constituent cells of an embryo sac are connected with each other through plasmodesmata. The outer wall of embryo sac is, however, devoid of plasmodesmata and is occasionally covered with a thin layer of cutin. The description of various constituent cells of a mature embryo sac and their functions are: first, the synergids. Synergids, also known as helpers, are attached to the micropylar end of the embryo sac by their pointed upper ends, and their rounded lower ends protrude into the embryo sac. If there are 2 or more synergids in an embryo sac, they almost completely embrace the egg cell on the lateral sides. The synergids also have a distinct wall around the micropylar 1/3 of the cell, which thins towards the callosal end and finally disappears. As a result, the callosal 1/3 of the cell lacks a wall. In epidendrum scutula, however, a distinct wall is present around the entire synergid. The synergid is marked by a prominent system of filiform apparatus consisting of many finger-like projections. These projections arise from the cell wall of the synergid and penetrate into the cytoplasm of the central cell. Electron microscopic studies have revealed that each finger-like projection represents well-packed microfibrils containing polysaccharides within a non-fibrillar sheath. The cytoplasm of the synergid is strongly polarized. The callosal region of the cell is occupied by one large or many small vacuoles. The micropylar region of the cell is occupied by a prominent nucleus and large amount of cytoplasm. The cytoplasm is rich in mitochondria, endoplasmic reticulum, and dictyosomes, which are especially concentrated near the filiform apparatus. Similarly, lipids, RNA, and proteins are also abundant and concentrated in this region. The connections between synergids and the central cell are mainly through plasmodesmata. Synergids have generally a limited period of usefulness and they break down after fertilization. Sometimes they may persist even after the entry of pollen tube. For example, in Trapilla, the synergids enlarge and remain active as haustoria even when the embryo is mature. In Astraceae, the synergids extend beyond the micropyle and functions as haustoria, and in the Santalaceae they form branched haustoria. The various functions which are attributed to synergids are: distribution of nutrients in the embryo sac, secretion of substances which attract pollen tube during fertilization. Degenerated synergids form suitable space for the pollen tube discharge and providing mechanical strength. Number 2, egg cell. The egg cell along with 2 synergids forms egg apparatus at the micropylar end of the embryo sac. The egg cell is partly embraced by 2 synergids on the lateral sides. The egg shows common walls with the 2 synergids and the central cell. It has a wall similar in structure and extent to the synergids. The wall is thicker in the micropylar region but becomes thinner towards the callosal end. In Capsula, Gossypium, Ziamase, and Taurinia, the egg has no wall towards the callosal end, and in Epidendron scutula, a uniformly thick wall is found all around the egg. The egg cell is connected with the synergids and central cell by plasmodesmata. As the egg matures, its polarity is established. The entire cytoplasm of the egg cell lies at the causal end, whereas the micropylar end is occupied by a large vacuole. In Ziamase and epidendron, however, the cytoplasm is evenly distributed throughout the cell. The nucleus of the egg cell is not markedly different from the synergid nucleus except for a slightly larger size. The egg cytoplasm contains plastids and ribosomes. Ultrastructural studies have revealed that mitochondria in the egg cell have fewer cristae and dictyosomes are either absent or very few in number. These features suggest passive nature of the egg cytoplasm. In plumbago and plumbagilla type embryo sacs, which lack synergids, the egg cell develops prominent projections at its micropylar end, which resembles the filiform outgrowths generally found in the synergids. It seems that in the absence of synergids, the egg cell itself takes over the role of synergids. Number 3, antipodal cells. These cells lie at the causal end of the embryo sac. They exhibit greatest variation amongst all the cells of the embryo sac. Usually, they degenerate before or soon after fertilization without any appreciable enlargement. In a typical polygonum type of embryo sac, there are 3 antipodals, but occasionally they may divide and then their number increases. In Ziamase, there are 20 antipodals, and in Sasa paniculata, there are as many as 300 antipodals. Multicellular antipodal complexes are found in the members of the Ranunculaceae, Papaveraceae, Apiaceae, Gentianaceae, Asteraceae, and Povaceae. Antipodals are altogether absent in Oenothera type of embryo sac. Generally, each antipodal cell has a distinct and definite cell wall, but in many families as Brassicaceae, Melastomaceae, Polygonaceae, Orchidaceae, and Sapotaceae, cell wall formation does not take place and the antipodal nuclei remain naked. The cytoplasm of the antipodal cell is rich in mitochondria, plastids, and dictyosomes. The vacuoles present in the cytoplasm probably originates from dictyosomes or endoplasmic reticulum. Histochemical investigations have shown that the cytoplasm of antipodals has high concentration of protein peroxidase, cytochrome oxidase, and ascorbic acid. Thus, histochemically, the cytoplasm of antipodals is very active and shows similarity with that of nutritive cells. Hence, antipodals are considered to be related with the nutrition of the embryo sac. Number 4, central cell. This is the largest cell of the embryo sac. It contains 2 polar nuclei which fuse before fertilization to form the secondary nucleus. The latter forms endosperm after fertilization. Hence, the central cell is also referred to as endosperm mother cell. The nuclei of the central cell are very large and each possesses a conspicuous nucleolus. There is considerable variation in the number of polar nuclei in an embryo sac. In 4 nucleate Oenothera type of embryo sac, there is only 1 polar nucleus, whereas Pinia and Plumbago type of embryo sacs have 3 to 4 polar nuclei. The cytoplasm of the central cell has numerous mitochondria, dictyosomes, ribosomes, and plastids. Although there is no connection between the central cell and the cells of the nucleus, the cytoplasm of the central cell is in contact with that of the egg cell synergids and antipodals through plasmodesmata. Dear students, now we will discuss the development of the female gametophyte. Female gametophyte development occurs over two phases referred to as Megasporogenesis and Megagametogenesis. More than 15 different patterns of female gametophyte development have been described. The different patterns arise mainly from variations in cytokinesis during meiosis, number and pattern of mitotic divisions, and pattern of cellularization. The developmental pattern exhibited by most species is referred to as the polygonum type because it was first described in Polygonum divaricatum. The polygonum type female gametophyte is exhibited by more than 70% of flowering plants and is the pattern found in many economically and biologically important groups including Brassicaceae, for example Arabidopsis, Capsula, Brassica, Gramineae, for example maize, rice, wheat, Malvaceae, for example cotton, Leguminosae, for example beans, soybean, and Solanaceae, for example pepper, tobacco, tomato, potato, and petunia. Now the various phases of female gametophyte. First, the megasporogenesis. During megasporogenesis, a single hypodermal cell in the nucleus functions as the archisporium. It becomes more prominent than its surrounding cells because of its large size, denser cytoplasm, and larger nucleus. In tenuinucleate and pseudocrasinucleate ovules, the archisporial cell directly functions as megaspore mother cell, whereas in crasinucleate ovules it divides periclinally, cutting an outer primary parietal cell and an inner primary sporogenous cell. The latter functions as the megaspore mother cell. During megasporogenesis, the diploid megaspore mother cell, also called megasporocyte, undergoes meiosis and gives rise to 4 haploid nuclei. Angiosperms exhibit 3 main patterns of megasporogenesis referred to as monosporic, bisporic, and tetrasporic. The 3 types differ mainly in whether cell plate formation occurs after these divisions, thus determining the number of meiotic products that contribute to the formation of the mature female gametophyte. In the monosporic pattern, both meiotic divisions are accompanied by cell plate formation, resulting in 4 one-nucleate megaspores. Subsequently, 3 megaspores, generally the micropylar, most megaspores undergo cell death. In the bisporic pattern, cell plate form after meiosis first, but not meiosis second. The result is two nucleate megaspores, one of which degenerates. In the tetrasporic pattern, cell plates fail to form after both meiotic divisions, resulting in 1 4-nucleate megaspores. Thus, these 3 patterns give rise to a single functional megaspore that contains 1 monosporic, 2 bisporic, or 4 tetrasporic meiotic nuclei. The monosporic pattern is the most common form and is represented within the polygonum pattern. Now the megagametogenesis. During megagametogenesis, the functional megaspore gives rise to the mature female gametophyte. Initially, the megaspore undergoes 1 or 2 rounds of mitosis without cytokinesis, resulting in a multinucleate cenocyte. Subsequently, cell wall form around these nuclei, resulting in a cellularized female gametophyte. For example, in the polygonum type pattern, a single nucleus undergoes 2 rounds of mitosis producing a 4-nucleate cell with 2 nuclei at each pole. During a third mitosis, phragmoplasts and cell plates form between sister and non-sister nuclei, and soon thereafter the female gametophyte cells become completely surrounded by cell walls. During cellularization, 2 nuclei, one from each pole, migrate towards the center of the developing female gametophyte and fuse together either before or upon fertilization of the central cell. These events result in a 7-celled structure consisting of 3 antipodal cells, 1 central cell, 2 synergid cells and 1 egg cell. The monosporic polygonum type of female gametophyte is typically a 7-celled structure at maturity. However, this structure may be modified by cell death or cell proliferation events in various species. For example, in Arabidopsis, the antipodal cells undergo cell death immediately before fertilization, whereas in grasses, for example in maize, the antipodal cells proliferate. Throughout development, the female gametophyte exhibits a polarity along its causal micropolar axis. During polygonum type Megasporogenesis, the causal most megaspore survives and the other 3 megaspores undergo cell death. During cell differentiation, the nuclei at the micropolar end becomes specified to develop into the egg cell, the micropolar polar nucleus and the synergid cells. The causal nuclei develop into the 3 antipodal cells and the causal polar nucleus. Furthermore, all of the cells within the female gametophyte differentiate into polar structures. For example, in many species, the egg cell's nucleus is located towards the causal end and its vacuole occupies the micropolar end. By contrast, the synergid and central cells have the opposite polarity. Thus, the establishment of polarity within the female gametophyte corresponds to the asymmetric development of the surrounding ovule layers, suggesting that female gametophyte polarity is regulated at least in part by the surrounding sporophytic tissues. Sporophytic factors that influence female gametophyte development have yet to be identified. Now, fertilization. Soon after pollen is transferred from anther to stigma, the male gametophyte forms a pollen tube that grows via a tip growth process through the carpel's sporophytic tissue to reach the female gametophyte. The pollen tube enters the female gametophyte by growing into one of the 2 synergid cells through a structurally elaborated portion of the micropolar cell wall known as the filiform apparatus. The penetrated synergid cell undergoes cell death soon before or upon pollen tube arrival. Immediately after arrival, pollen tube growth ceases. An aperture forms at or near the pollen tube tip, and the contents of the pollen tube, including the 2 sperm cells, are released rapidly into the degenerating synergid cytoplasm. Double fertilization occurs when the 2 sperm cells migrate to the egg and central cells and their plasma membranes fuse with their respective target cell to transport the sperm nuclei for karyogamy. Synergid cell death may be a prerequisite for normal fertilization and angiosperms. For example, the process of degeneration itself could decrease resistance to both pollen tube penetration and sperm cell migration during fertilization. In addition, synergid degeneration generally is accompanied by cytoskeletal reorganization that may facilitate male gamete transfer from the pollen tube to the egg and the central cell. Dear students, this is all about structure of carpel and female gametophyte. Dear students, we have discussed over here structure of carpel and female gametophyte. We have also discussed here the development of female gametophyte and fertilization. Thank you very much. God bless you.